Good afternoon, children. Today I'm going to explain about biomolecules chapter 4 marks questions. So the first 4 mark question is write a brief account on polysaccharides. So actually the polysaccharide nothing but those are sugar molecules. So those sugar molecules are attached with each other. So to form a long chain-like structure of the sugar molecule, so which is called as polysaccharides. So actually these polysaccharides are acid insoluble carbohydrate carbohydrate macromolecules. So actually it is mainly made up with that a long chain of sugars. So this is the first point regarding to it. So all the polysaccharides are mainly composed with a long chain of sugars. So for example, there is a glucose molecule. The glucose molecule are attached with the another glucose molecules by the glycosidic bond. So many number of the glucose molecules are attached with each other. So to form a long chain. So that nothing but it is called as a polysaccharide. Okay, actually thread containing different monosaccharides as building blocks. So example, cotton thread. So actually the cotton thread is mainly made up with the cellulosic substances. So cellulose is one type of polysaccharide. So just you see that. So here the different type of polysaccharides are made up the different building blocks for examples. So here the cellulose, so that mainly mainly made up with that glucose monomer. So here many number of the glucose molecules so that are attached with the one more glucose molecules to form a long chain. So these long chain of glucose monomers that constitute the cellulose. So where has come to that insulin? So many number of the fructose molecules are arranged in a long chain. So that's why the fructose monomers compose the insulin. So next term that is a glycogen. So glycogen means so glucose monomers. Okay, so these are the different type of polysaccharides that are made up of the different building blocks. So here, so the various type of the polysaccharides are present in the organism. So for example, the glycogen, so it is a reserve food material that present in the animals, and whereas compared to the chitin, so actually it is an exoskeleton of arthropods and cell wall of the fungi. So here the cell wall of the fungi especially made up of the chitin. So chitin is in a one type of polysaccharides. So in animals, the main material, main carbohydrate material is in a glycogen. So glycogen is in a one type of polysaccharides. So next one, N-acetyl glucosamine. So it is in a bacterial cell wall is especially made up with that N-acetyl glucosamine. So it is in a one type of polysaccharide. Okay. So actually the polysaccharide molecules are source as an energy reservoirs because of so these molecules are involved in the cellular respirations to release the energy. So that's why they are act as an energy reservoirs because of they store the energy and they release the energy during the cellular respiration. Okay, so you have the cellulosic cell wall and external protein protections like fungi, plant, and exoskeleton of orthopods that mainly made up with the polysaccharides. So these are the points that are mainly regarding to the polysaccharides. Okay, so coming to the next question, the schematically represented primary, secondary, tertiary structure of the hypothetical polymer using protein example. So actually, so the proteins are also biomolecules, so which are mainly or abundantly found in the both animals and plants. Okay, so these are biological macromolecules like proteins. So undergo into a folding process through which they're biologically active and form is produced. So it is in a sequential folding in the result of the form. Okay, so whatever the protein when it is formed during that protein synthesis by the ribosomes. Okay, so actually the ribosomes produce the proteins in the different forms. That is in a primary, and second one is in a secondary, third one is in a tertiary, and fourth one is in a quaternary protein. Okay, so while the protein synthesis takes place by the ribosome, so just to see that there are many number of the amino acids because of so whatever the protein which is mainly constituted with the many number of the amino acids that are attached with the peptide bonds. So the amino acids are attached with each other by the peptide bond. Just to see here, so it is an serine, one amino acid that attached with the cysteine. The cysteine amino acid that attached with the tyrosine, and tyrosine amino acid attached with the glutamic acid. So these only amino acids are arranged in a chain-like structure. So this chain-like arrangement of the amino acids, that nothing but it is in a primary structure of the protein. Primary structure of the protein. So whereas come to that secondary structure. So secondary structure of the protein, that especially shows helical form. Okay, so whatever the primary, so it found in the form of linear sequence of the amino acids. So which means many number of the amino acids are arranged in a long chain like structure. So that's why, so it is in a linear sequence of amino acids. So first amino acid that present in the protein, that nothing but it is called as an N-terminal amino acid, and whatever the last amino acid is called as C-terminal amino acids. Okay, so in a linear sequence, so the first one is called as N-terminal amino acid, and the last one is called as C-terminal amino acid. So whereas come to that secondary structure of the protein, so whatever the polypeptide chain, so which has an N-terminal and C-terminal amino acids, so that undergo mainly the folding. Okay, the folding amino, all the folding polypeptide chain, so which leads to the formation of the secondary structure of the proteins. So which is mainly stabilized by the hydrogen bond. So mainly the right-handed helices are observed. So the right-handed helices are observed in the secondary structure of the protein. So that nothing but it is called as in a helical structure. So whereas come to the tertiary structure of the protein, so whatever the long protein chain is folding upon itself, long protein chain folding upon itself, to like a hollow woolen like ball. So whenever we take the thread of the woolen and we can round it the woolen in a ball like structures. So that ball like structure of the long chain of the protein, so that nothing but it is called as a tertiary structure of the protein. So these tertiary structure of the proteins that given a three-dimensional view of the proteins, example is a myosin. Okay, so now the third question is nucleic acids exhibit secondary structure. So justify with one example. So it is also very important for mass question. So actually the nucleic acids exhibit the wide variety of the secondary structure. So for example, secondary structure of the DNA is in a famous model that was proposed by the Watson and Crick scientists. So actually the Watson and Crick scientists that proposed the helical model, okay, helical structure of the DNA. So according to this model, the DNA exists in double helix. So just you see the figure here, the two helical structure threads are attached with the each other, but nothing but it form in a double helix model. Double helix model. So the two polynucleotide strands are anti-parallel. The backbone is formed by the sugar phosphate and sugar chain. Okay, so just you see that. So actually this is a one thread of the DNA, another thread of the DNA. So these both threads are both polynucleated strands are arranged anti-parallel. So whatever that, so these nucleotide that mainly contain three parts. One is in a nitrogen base pair, and second one is in a sugar molecules, and third one is in a phosphorus molecule. Okay, so these phosphorus and sugars that act as in a backbone. Okay, so backbone for the nucleotide. So whatever the nitrogen base pairs, so that are projected perpendicular to backbone of the facing inside. So just you see here, it is in a nitrogen base pair guanine, and it is in a nitrogen base pair cyanine. Okay, so cytosine. Okay, so guanine and cytosine. So nitrogen bases are there are many. There are only the four nitrogen base pairs are there. There are adenine and thymine and guanine and cytosine. So here the adenine that represented with the A capital letter, and it is always combined with the thymine, so which represents with the T. So by the two hydrogen bonds. So whereas compared to the guanine, so it is in a nitrogen base pairs represents with the G's and combined with the cytosine C by the three hydrogen bonds. So here one important point to be remember. So always the adenine nitrogen base pairs is combined with the thymine only, and always the guanine combines with the cytosine only. Okay, so here one full turn of that helical strands, so would involves in the 10 nitrogen base pairs. So there are 10 nitrogen base pairs are present in the one helical strand. So here the length of the one coil is 34 angstrom. So that's why the distance between the one base pair is in a 3.4 angstrom. So because of it, each helical strand that mainly contain 10 base pairs. So the length of the helical strand is 34 angstrom. That's why the 34 by 10 that equals to the 3.4 angstroms. So in the distance between the 3.4 angstrom, there are many number of the base pairs that are present in the nucleotide. So this type of DNA normally it is called as a bDNA. So it is called as a bDNA. Okay, so these are the points that are mainly regarding to that nucleic acids exhibit secondary structures. So whatever that three questions I explained in this session. So everybody uh, should learn these three questions yourself, and thank you.